It's beautiful out as it looks. Nice and warm. Okay. 22 years ago. I remember that. Wow. We had a 9-month-old. Kind of wondered what the world was, what the future going to be like for you guys. Anyway, these verses help. Look up to the mountains, because my help comes from there. My help comes from the Lord and the heavenly rivers. You guys have lived after the changes. You go through those changes 22 years ago. It was right. So let's open the word of prayer. Father, thank you so much for a beautiful day. Thank you for the time to reflect and remember what happened 22 years ago. The day for many of us that the world changed. Father, I pray that for those who are still struggling with the loss, struggling with the effects of what happened in New York and Washington, Pennsylvania so long ago, I pray that they would find some comfort and peace. Father, I ask that all of us this week would continue to work diligently to keep in mind the plans and will that you have for our lives. Father, I pray that all of us would continue to look to you for help. If we are not certain of how to do that, we would ask those who can assist. Father, thank you so much for your students here, especially those who love and support. All right, so we are on to cells today. This is the last of the topics, which means that Friday we are ready for the exam. Do not forget that Thursday night is a review session. I have asked Noah, who were at the review session, to try to do that as a virtual setup, or at least record it, so that some of you who cannot be there will be able to view it. How many of you were there last Thursday? You. I would encourage you to go, especially if you have questions. Go prepared to ask questions. Don't go just to sit and let him speak at you. But make sure that you are going to say, hey, I don't understand this, This is what I understand. Is that correct? Whatever the case may be. Wednesday, it's very possible we'll have a little bit of a review time. So come on Wednesday with some questions. So that's what's going on. We have 3 quizzes now available, or I'll modify the cells here very soon, so that's available. And then we are doing a diffusion and osmosis lab this week. So come prepared to learn about how molecules move across membranes. It's exciting stuff. It really is. And it's very fundamental to what we're going to be doing the rest of this class time, or the whole class. Okay? All right, that's for the last time, so I'm moving through. So we have been down here with the atoms, remember, and we have moved up to molecules. We've finished the discussion of those molecules and the macromolecules. Today we are going to put organelles in the cell. We'll talk about the big cell, and then we'll move it down to the smaller organelles. Okay, so that's the order in which we expect to go today. So when we were together last time, remember that we were building polymers. Just to reiterate how that process occurs. It is a dehydration reaction, which means that 2 building blocks, what we call monomers, are added together. So if we're talking carbohydrates, one of our monomers is glucose. If we're talking proteins, it's amino acids. If we're talking lipids, it is fatty acids. If we were talking about nucleic acids, it's nucleotides. It doesn't matter. They get put together in the same kind of reaction. Dehydration. Dehydration, which means that water is a product. Always, water is a product. Now, that reaction is going to require energy. So we also describe it as one that is endergonic. Energy needs to be put in. Okay, and then the other word there is synthesis. So it is anabolic. It is building up. However you get there, that's what's going on. Okay? Now, once we have built this large molecule, that large molecule can be split into the individual units. The process there is called hydrolysis. Water splitting. That's what that word means. So we can see that water is added to that bond, and we have the two monomers again. Talking carbohydrates, starch, if you are a plant, or glycogen, if you are an animal. That's your storage form, that's what you're going to break down. If we're talking nucleic acids, we break it down to nucleotides. If we're talking proteins, we break it down to amino acids. Talking about a triglyceride, we cleave off fatty acids. Same process, it doesn't matter, just different building blocks. And it is hydrolysis, as I said, energy will be released, so it is exergonic. It is breaking down, so it is catabolic. Any of those ways of getting there, okay, that's the point. Now, we then moved on and talked a little bit about proteins, which we said was a little complicated. 4 levels of organization. All proteins have the first 3. Only some have the 4th. So all have primary sequence, just simply what is the sequence of amino acids. Those are then going to have interactions with each other and create some of these weird-looking shapes called helices and beta sheets. I'm not going to ask that, but that's what they are in a second. That molecule does not exist on its own. It's in water. Because, again, water is our solvent. So it folds, it twists, it bends, it does all sorts of things because some amino acids like water and others do not. So the ones that like water go out to the outside. The ones that don't get folded into the center. That becomes the structure that is functional. It is what's going to carry out the function that protein will have. Change to primary means that the secondary might change, which means the tertiary might change, which means the protein doesn't work the way it should. Change the conditions that the tertiary is found in, this functional molecule, it may not function the way it ought to. That's why we concern ourselves with blood pH and other things like that. We'll get there. Okay, that's down the road. Quaternary, that's the 4th level, and it is an assembly of several subunits. Not every protein has that level. Hemoglobin is one. All right, then we talked about a specialization of proteins, and that's called an enzyme. Okay? Now, do not assume that all enzymes in biology are proteins. There is a few exceptions to that. That, if you were told that in high school, that's not quite right. The majority are, like 99%, but there are a few exceptions. So what does an enzyme do? It finds a substrate. That's the reactant. That's what's going to be worked upon. And it has an active site where that substrate fits. And it is very specific. I mentioned on Friday, this is sucrose. We know the other disaccharides are lactose, maltose. Is it in there? No. Can that enzyme work on it? No. We have individual enzymes for lactose and maltose. This one is sucrase or sucrose. So when it fits into the active site, it's as if things kind of twist it a little bit or cause it to not be in its nice, stable condition situation. And when that happens, boom, you get the products. What happens to the enzyme? Nothing. It's ready to go again and again and again and again. You don't need a lot of enzyme to carry out a lot of reactions. That's the beautiful thing about enzymes. All right, that's where we finished last time. Any questions? Yeah.
So on the written exam review sheet, it says one of an enzyme and then it says characteristics of an enzyme. So would that be that it can continue to grow?
That would be one. Usually that it's protein. That would be something else that you could file away for characteristics. But yes, that they are reusable. If they don't change, that definitely would be enough. Anything else? All right. So, there is the typical cell. Typical animal cell. Plant cells look very different. So this is a typical animal cell. The artist drew it so that it has everything a cell might possibly have in it. Do all cells have absolutely everything that a cell may have? No, not always. But we notice a few things about this cell. There is something called a nucleus here. There's this structure that looks really weird called endoplasmic reticulum. There are these little squiggly structures called mitochondria. If I say mitochondria, you say, yeah, you always say that. Powerhouse cell. Does so much, it does other things, but that's what you always remember. Okay? And there's some little droplets here that are called vesicles. Vesicles are the transports. And we see, let's see, where is it? I don't see it. It's up there. Oh, there it is. The green stuff right in front of me. That's called the Golgi. It's either apparatus or complex. It goes by different names. We don't have another name for it. Golgi is an actual person, and we're trying to get away from calling things by people's names, but nobody's come up with anything else. So there you go. So this is kind of the packaging center. Right, so things that are going to be packaged into these vesicles, they go through the Golgi. We'll get into a little bit more of that later. Little dots all over the place, those are ribosomes. Little thin lines that you're seeing all over the place, that's the cytoskeleton. Cells have cytoskeleton, otherwise they wouldn't have a good shape or the shape they're supposed to. Now this is a epithelial cell. Epithelial cells are one of the 4 tissue types that we'll talk about starting next Monday. Epithelial cells are on the surface of everything that you have in your body. Epithelial cells in your mouth. Epithelial cells in your blood vessels. Epithelial cells on the outside of your kidneys. Every surface has an epithelial tissue. The reason we point that out is because it has basal, that's where it's the bottom. It has apical, that's the outside or the top. And it has laterals, okay? So it's just kind of a way to orient this cell. All right, so there's other features up there that we're going to get to in a little bit. But this is typical. So when we say cells, what is a cell? All right, this very first one. You ready? You're going to write this down. You're going to remember it. You're going to get it right on the exam. This is here that everybody gets this question right. It's never happened. What is a cell? A cell is the smallest, simplest structural and functional unit of life. I am giving you the answer to the question. Literally, I am giving you the answer to a question. Now, I'm not going to state it so bluntly. What is a cell? But you're going to get the answer this year, right? 100% of you will get it right. It's never happened. Never. Never. They don't know why. They tell you there's the answer to a question. Maybe it's because the people get it wrong out here right now. I don't know, but what is a cell? Smallest, simplest, structural and functional unit of life. If you're not made of cells, then what is true? You're not living. It's as simple as that. Viruses, not living, not made of cells. Don't let the Lysol bottle tell you otherwise. Because it says kills 99% of the viruses. You can't kill a virus if it's not living. Right? Okay? Smallest, simplest, structural, functional unit of life. So those organelles that we just briefly talked about, like the nucleus, the endoplasmic reticulum, the mitochondria, are those living? No, they're in the cell. Therefore, they are not living. They're part of the cell. Because, hey, what? Guess what? Next year, if you're in nursing, you take microbiology, and there you're going to learn there are some cells that don't have organelles. Bacteria, they're still living. They're a structural, functional unit of life. They are a cell. Okay? Smallest, simplest, structural, functional unit of life. Everybody's going to get it right this year. Therefore, all our organisms are composed of cells and cell products, especially if you're multicellular. You have cell products. Your tendons, your ligaments, bone material, that's your bone, blood material, that's your blood. All of those are products of cells. Cells of your body make them. Okay? If you're a single cell, you don't really have cell products. An organism, I should make that possessive. An organism's structure and function is dependent upon the activities of its cells. Why do we go and look at and talk about cells? Because if something's wrong with the cell, it could damage or harm you as an organ. Diseases ultimately come down to cellular responses. They're not functional, you're not functional. Something's gone awry. Okay, so go down to this basic level. And if you're going to go on and do pharmacology, how does the drug get into the cell? When it gets into the cell, what does it do to the cell? There's understandings of the cell. You've got to have it. Cells come only from pre-existing cells. None of this spontaneous generation stuff that was talked about back in the 1700s and 1800s. Well, remember Louis Pasteur? He was the one that helped repeat that. And they used to think that, a fly is really sitting there, flies suddenly doesn't have a right to live. Oh my gosh. Getting a little aggressive, Nora. I mean, oh well. It's alright. If you remember rotting meat and flies suddenly appeared and they thought that they just spontaneously generated out of meat. They didn't know there were eggs in the meat. They became larvae and became flies. Cells come only from pre-existing cells. And that means cells divide and become new cells. And then lastly, which is important for study, or especially research purposes, cells from different organisms have similar chemical composition and metabolic mechanisms. We have used the proverbial lab rat, guinea pig, in research for many, many years, because we know that a mouse has similar chemical properties, composition, and metabolic mechanism. You give a mouse a drug, and we can expect that drug to do something similar in us. That's the whole idea. Whether you like it or not, experimental animals have given us most of our medicine and most of our understanding of medicine. So, important. Also important when we think about insulin and other drugs that bacteria make for us. The whole reason that works is because of this item. We manufacture or design bacteria to make insulin. All right, so what is a cell? Smallest, simplest, structural, functional unit of life. If that is true, then the second one is true. All right, and it can be structural and functional. You change the structure, you change the function, and the cell changes the way it's going to behave, which could have effects. All linked together. All right. So, in the human body, we describe cells based on shape very often, especially the epithelial tissues that I was just talking about. So there are several shapes which you are going to see and be described to you many, many times. First is squamous. Squamous simply means that it's a flat cell. I'll show you a picture in a second. All right, but if you think pancake, you're right on target. Second, cuboidal. Shaped like a cube. Yeah, shaped like a cube. As tall as it is wide. Columnar. Columnar are in columns. Are they Doric or Ionic? We don't care, they're just called instead of as wide. Now the reason that we emphasize these shapes and a few others that are coming up, but these three to start, each shape means that they have a different function. Okay? Each shape means that they're going to have a different function. So they're going to be in different places throughout the body because they have a specific function. And if the shape changes, that could change the function. For instance, you have a tissue in your trachea, in your bronchi, that is a columnar cell. If you were to ingest certain chemicals through breathing, i.e. smoking, those cells may change. Instead of being columnar, they become cuboidal. They will function the same way. That's why we are interested in cell shapes. Now, we have other cells. These are not what we would find in the epithelial tissues, the surface layer tissues or cells. We have spheroid, of course, your blood cells. So, those are kind of the four. And as we just mentioned, we know that these cells, if they're epithelial, have these borders, boundaries that we can describe. If it's basal, we say it's at the bottom. It's what it is, the bottom surface, and it rests on something called a basement membrane. It's what's underneath it. Laterals, of course, are the sides. Squamous cell really doesn't have much of a lateral border. Cuboidal and columnar shape. And then apical is the upper surface, outer surface. Obviously, if I'm saying exposed to fluid, and I'm saying your skin is also an epithelial tissue that's just outside the body. So, there's our shapes. So this yellow line here, in these first three, yellow line down there, that's the basement membrane that I just mentioned. That's what the cells are left. You can see that the squamous cells, again, really don't have lateral sides. But they certainly have an apical and a basal. Apical, basal, laterals. Apical, basal, laterals. Okay? Then we get down here into all these other funky, weird shapes. And of course, they're not resting on something, so which way's up and which way's down is not really relevant. And eventually, we'll talk about things that are star-shaped and different shapes like this. But those aren't that common. They're specific to certain tissues. All right, so there's our typical cell again. Basal, lateral, apical. I want you to be familiar with those terms because we're going to, as we go through the course, not just this week, but all the way through to next May, I will talk about things that are apical or basal, etc. So familiarization with this is going to be important. All right. Now, that cell surface, what's creating the border to the cell is called the plasma membrane. Plasma membrane. The plasma membrane is composed of phospholipids. So that specialized lipid that we were talking about last week. We mentioned lipids, and the first one I mentioned was the triglyceride. Glycerol with 3 lipids, 3 fatty acids attached to it. And then I said that there's some that have only 2 fatty acids. And the additional position is occupied by a phosphate. Now, this is a bilayer, which means it's 2 layers. So this artist's representation is what we is our best understanding. So here, the little squiggles, remember those are fatty acids. The little balls, those are the phosphate groups. So we call these the heads and these the tails. Now, the phosphate group, remember, is ionized. It has a charge to it. So it wants to be near water. There's water here, there's water here. There's water outside the cell, there's water inside the cell. So the phosphate groups need to be on the surface. And the fatty acids, well, they don't like water, so they're in the center, away from the water. Now, what's the big idea about that? If you are an ion, say you're a sodium ion, you're charged, you're here in the water, and some way, somehow, you need to get inside the cell. Well, can you pass through where there is no water? Nope. Ions can't move through that lipid environment. Because lipids, remember, do not mix with, and if anything's dissolved in water, they can't mix with the lipids either. So, we have to have special ways to get things from one side to the other if they are not lipid soluble. They don't like lipids. The other takeaway, notice these are very tightly packed. Okay, there's no space between them. So if you're too large, well, you're not getting through either. And if you've heard the term semi-permeable, that's what we're describing. Certain things get through, other things do not. Permeable means the ability to move through. So you're either fully permeable, semi-permeable, or impermeable, meaning you can't get anything through. So these are semi, because if you're a lipid, glycine molecule, you can go through. You are an ion, you are Gandalf. Greatest book series ever. Tell me that what's her face's books are better. It's not. So, that's what we're talking about with this phospholipid bilayer. Oh, and one other cool thing, if you're into chemistry. If you were to make these things and drop them in the water, they make a circle automatically. And they put their tails just like that. They do this automatically. It's so cool. No energy involved. They just do it because of the chemical. Alright. So, when we describe this plasma membrane, we know it's made of phospholipids, and you just notice there's some things embedded in the membrane. It's not just phospholipids. There's other stuff in there. And so our best description is what's called the fluid mosaic model. Meaning, first of all, fluid. It's not rigid. Okay? It's not rigid. It has fluidity. Okay? Things can move in our phospholipid. Means it's kind of pliable. Which is good, right? Because if you have really rigid cells like plants, then you're like a plant. You're not going to move. So that's good. And it means it's mosaic. What the heck is that? Well, you have to think about the tile floors that people do and did. Mosaics. Things are randomly designed. Okay? So it looks like that. So we have our phospholipids, 1 layer, 2 layer, extracellular layer, intracellular layer, lipids, phosphate groups. And then embedded in it are some proteins. Okay. They're either going through it, meaning that they span transmembrane, or they're on the periphery, like these. And it's not just proteins, there's also cholesterol. Remember I said you need cholesterol in your diet? Here's another reason why, not just steroids. Cholesterol is embedded in the cell membrane, helps give it fluidity. And there are carbohydrates attached to things. It's called a glycolipid because it has a carbohydrate attached to it. Or there should be somewhere, not on this one, sometimes we have lipids attached to proteins as well. So we have multiple things in this cell membrane. And if I were to tag this particular glycoprotein in some way so I could recognize it, come back a couple hours later, it may not be in the same place, it may have moved. That's the fluidity. Alright, so those membranes are not just phospholipids, as I mentioned, also cholesterol, and then glycolipids, carbohydrates attached to lipids. Why are those key? Because part of, in some way, glycolipids help create a cell identity. That's what I'm trying to say. So your cells are your cells. Your red blood cells are your red blood cells because, in some cases, glycolipids. They mark the cell. The membrane proteins, as I mentioned, integral, as in they're embedded, or peripheral rather. They're on one side of the membrane. They're one layer of the membrane. And transmembrane go all the way through the membrane. Okay, so they're from one side to the other. Now, if we think about these transmembranes, we think about these transmembranes, as we see this protein going all the way through, we see that a part of it is outside and a part of it is inside. water, this is water. So what must be true of the amino acids of that protein? Hydrophilic or hydrophobic? Do they like water or do they not like water? They're right here on the surface. They have to like water, okay, because they're in contact with water. What about the amino acids that are here, going through the lipids? Hydrophobic. Very, very important in our understanding. But that means these have to be hydrophilic. They have to like water because they're in contact with it. These have to be hydrophobic because they're in contact with lipids. Now, this particular protein is called a channel. And guess what? You can see right through there. There's a channel. There's an opening. What are the amino acids in here? Hydrophilic or hydrophobic? Did I hear phobic? Philic. Because if it's open like that, water goes in. So that means inside of that channel, it also has to be hydrophilic. It has to like water because it's in contact with water. Remember we said, how's an ion that is dissolved in water from here to here and here? Oh. Might have just clued you in a little bit. Channel. Way to get through. But that's the idea. All right. And there's our hydrophobic and hydrophobic. I had it up there again. So these are the hydrophobics. These are the hydrophilic areas. You're going to take cell biology next semester. Some of you are. There's the alpha helices spanning the membrane. FYI for next semester. All right. So when we talk about this cell surface, we said that there's some proteins embedded in it. And those proteins are going to have specific roles. Different cells will have different proteins embedded in it. One role is to be a receptor for hormones, among other things. So last time we were together, we talked about insulin as a hormone, it's a protein. And it has to cause cells to take glucose out of the blood and store it in the cell. Well, what's going to signal the cell to take glucose into the cell? The hormone insulin. Well, how's that going to cause the cell to do it? It has to bind to a receptor. A specific receptor for something to be a signal. Is it like an enzyme? Absolutely. It's that specific. Specific receptors for specific hormones. The next one is that it can act as an enzyme. We've already talked about enzymes. And lining your digestive tract, your small intestine specifically, that enzyme sucrase, the enzyme lactase, the enzyme maltase that I previously mentioned. All are enzymes embedded in the cell membranes of your body. So very, very important. There's the channel we just mentioned. There's an ion, water soluble. To get through this lipid environment of the cell membrane, it uses a channel where water can move through. So that's 3. But wait, there's more. Everybody good? Yeah. Yes. Yes, absolutely. Sure. Peripheral. Someone's through to something. Yes, good. By the way, these are all in the PowerPoint. Always look in the PowerPoint. Future reference. Not that I say what right in the front. Done? Is it almost? I'm sure. What? All right. Now, the other roles are carriers, motor molecules, cell identity, cell adhesion. And I got a picture of that. So these channels, again, just another view of the channels. Cell identity, as I mentioned, there's glycolipids, glycoproteins. These tell us what your cell is. Where is it in the body? Who does it belong to? You can't just take a kidney from anybody and give it to anybody. You have to match it. How do we know whether it's the right kidney? These things. You're able to show the right kidney. So it is cell identity. Why is your blood type A or O or AB? Cell identity. All these things are identifying cells. They also act in this case, or in this situation, as a CAM, it's called a cell adhesion molecule. Your skin remains intact because the cells are linked together by molecules, proteins that hold them together. They just happen. You certainly don't have glue holding your cells together or cement or anything else. These linking proteins that are binding one to another. Now, the other one that is not on the picture is a carrier molecule. Remember I said some things are too big to get through. How do you get them across? You attach to a protein, and the protein physically moves them across the membrane. So that's another structure. Or another protein function. The last one is the second messenger. I'll show a picture of that. Can I get him there? So if I go back, yeah, I got them all. Oh, motors! I forgot about motors. Motors, they make things move. I'll show an example of that. All right. So the last, these things, second messengers. Here, there are specialized proteins that are identified here as key proteins, and they cause the production of a messenger inside the cell. In other words, this receptor finds a messenger, but that messenger can get through the cell. It's like that's the gate. You knock on the door and hand the message to the gatekeeper, and the gatekeeper takes the message through and on to whomever it's delivered to. Well, in this case, that second messenger creates something called the adenylate cyclase. Don't worry about it. It's not on the exam. Just erase whatever you wrote down. Just mentioning it. It will come up again in this class, and it certainly will come up again in pharmacology. A lot of our drugs work on this system. All right. There are other surface projections, the apical layer, than just our cell membrane and the proteins embedded within. There are also things that are creating a little ruffle, folds, ruffles. These are extensions of the plasma membrane. Now the term Villi, singular villus, finger. Villi, fingers, little fingers. Cell membrane has ruffles. So when you look at it under the light microscope, it's right here, this little section right there. You look under an electron microscope and you can see, wow, those look like little fingers projected. Another view of it. And if you cut them, that's what they look like. So they're on the apical surface, and they're also called the brush border. When they looked at them under the light microscope, they said, oh, that looks like this was on the brush. Let's call it the brush border. Yeah. Now, they're predominantly in your small intestine. There's other areas, but that's a main location. So what's the function of these bristles, of these little finger-like projections? What's going on in the small intestine? Why do I need these? Why would you choose these? Catches the nutrients. Right idea. What do you do in the small intestine? Catch the nutrients. Give me another way of thinking about it. Absorb. And in order to help absorb things, what would be better? A flat cell or a cell with ruffles? Increase the surface area. You have more surface area, more things can move through. Good idea. So that's what microvilli do. Create more surface area. All right. Cilia. Cilia are single projections. They are not a part of the cell membrane. They are proteins that are projecting out of the cell. Most cells have between 20 to 200 if they have them. They beat in coordinated waves. So they all move and they reset. They all move and they reset. Just like being in the ocean. They are found in your upper respiratory tract. That's with everybody. Some of you have them in your oviducts. The rest of us have them in our inner ears. Function. What do they do? Think upper respiratory. What are they doing in there? Bronchi, trachea. What's their role? Not the air. Air's going to move on pressure. We'll talk about that next week. Good job. Yeah. They help expel mucus and everything like that. So it looks like that. Parts through that. There's your mucus. Trucking along. Give me a couple minutes. Hang on. Flagellum. One single projection in the human body. What cell has a flagellum? Used to ask way back when I was a naive teacher. Flagellum only found in the male's body. True or false? Well, that's false. I mean, that's true. And then somebody counters and says, no, it's false. Found in the women's body too sometimes. I wasn't thinking about that. All right, I'm showing you this picture, and you're going to want to copy that down. These are the organelles that you are going to need to know for the exam. These and only these. It's not an exhaustive list. There's some things that are organelles that are not on the list. But these are the ones I want you to know. And I am going to trust that you will learn them all you want without me having to simply tell you that the mitochondria are the... Okay? Just need that brief of an understanding. I'm not asking for volumes that Wikipedia will write for you. Just the kernel of truth about each one of these. Okay?